So, hello and welcome back to Excelsior Academy. So, guys, today now we are going to start with translation. So, we have done with the DNA replication, DNA repair, transcription, and now translation. So, this is also one of the most important topic as part of your CSI syllabus also. And अब तक हमने जितने भी proteins पढ़े थे, you can just imagine 10 times more. Excuse me. You have to remember because in this, whether it is prokaryotes or eukaryotes, it has got several protein factors which is required in order to carry out the translation. ठीक है? The first thing is like हमें पहले nucleoplasm से mRNA लाना है to your cytoplasm. For that also you need to know about the import and export of the proteins, and then coming to the cytoplasm, all about your mRNA, rRNA, and tRNA. Because now in the translation, all different types of your RNA are involved. ठीक है? So it is going to be very tricky. Plus, you have to remember a lot. Once you are able to remember, then only it is easy to understand the concepts. Okay, so without worrying, we'll do slowly, but we'll finish the translation in a simple manner. Okay, so let's start with our translation. So first thing what we are going to discuss over here is about the mRNA, the messenger RNA. So here I have taken all the important points only. So already we know what is mRNA, kaise bana, right? So only basic, the important we are going to discuss. So we all know what is open reading frame. So open reading frames क्या रहता है जहां से your ribosomes are going to read so that it is going to code for the amino acids. ठीक है? So the codons, it will be read in the form of the codons, and these will be giving information like which amino acids to be brought so that the polypeptide chain can be synthesized. Okay. So the translation machinery decodes only a portion of each mRNA. The information for protein synthesis is in the form of 3-letter code, that is your codons, which each specifies for one amino acid. So if you guys remember the genetic code, so there are, how many codons are there? Anyone can tell me like how many codons are there? कुछ idea है कितने codons हैं? Yes, anyone? So if you have seen the genetic code in which they have made, so you have seen like ऐसे U C A G करके होगा, and यहाँ पे भी same way U C A G, and in each also you will have U C A G. So यहाँ से भी आप calculate कर सकते हैं. So what they have done, so अगर आपको याद नहीं आ रहा है कितने codons हैं, so you can just calculate like this also. So how many, like what are the chances that here the probability of each the nucleotide can come over here? What is the probability over here? If I'm taking UCAG, okay, instead of T I'm taking U, तो हमारे पास 4 nucleotide है. So क्या, what is the combination over here that nucleotide can come? कितने nucleotides यहाँ पे आ सकते हैं in the first position? So you know codons are made up of 3 nucleotides, right? So यहाँ पे first position में कौन कौन से nucleotide आ सकते हैं?
मैम U.
No, the question is first position में, second position में, third position, right? So first position में क्या क्या आ सकता है? I'm asking. So out of 4, any 4 can come, correct?
Yes.
If I say what is the probability of the nucleotide coming in this position, then you can say that it is 1/4. That any one can come out of 4, any one can come, correct? अभी यहाँ पे, like in the case of second position, how many nucleotides can come if one has been used over here? Again 4, and similarly over here also. So 4 into 4 into 4 कितना हुआ?
64.
64. So you are going to have 64 combinations. Okay, so genetic code में 64 are there, and in our system also we have got the 64 codons. ठीक है? So अभी इतना clear है. अगर आपसे कोई probability पूछता है, so that is going to be 1/4. Unless until it says that कि यहाँ पे सिर्फ आपका pyrimidine आ सकता है या फिर purine आ सकता है. So then the probability becomes 1/2 because we have got 2 purines out of 4 and 2 pyrimidines out of 4. So आपकी probability of purine and pyrimidine becomes 1/2. और अगर कोई nucleotide पूछता है, then it is 1/4. I hope this much is clear, right? So this concept is clear because इसी पे based numericals भी होते हैं. Correct? Now, I don't know how much you guys will be remembering, but what I used to do is that in order to remember, एक तो हो गया आपके stop codons, ठीक है? So there are 3 stop codons are there, and there is one your start codon. And what you can do is you can just remember like जो आपके repeated nucleotides होते हैं, like all U, all C, all G, and all A. So if you could remember this also, आपको थोड़ा easy हो जाएगा when you are doing translation. So what is all U? It codes for phenylalanine. Okay. Now if it is all C, then it is proline. Now if it is all G, then I guess it is glycine. Sorry, G is over here, glycine. And if it is all A, then I guess it is your lysine. ये जहां तक मुझे याद है. If I'm wrong, just let me know later, okay? So if you could remember this also, more than enough. Okay, so let's continue now. So the protein coding regions of each mRNA is composed of contiguous non-overlapping string of codons called open reading frame, which is known as commonly ORFs. Now, in most of the cases, आपको भी ऐसे दिखेगा. So you know what is UTR region, you know what is CDS region. ठीक है, UTR क्या हो गया आपका untranslated region. In the case of the transcription, we have seen 5' UTR and 3' UTR, correct? CDS क्या हो गया आपका coding sequences. Okay, it is coding sequences. So this is the region which gets translated to your protein or amino acid polypeptide chain. Okay. Now each ORF specifies a single protein and starts and ends at internal sites within the mRNA. So that is the ends of the ORF are distinct from the ends of the mRNA. तो इसका क्या मतलब हुआ? ये अगर आपका mRNA है, so the codons in here is going to determine the end of the translation. यानी कि ORF का end होगा. So it is not necessary कि आपका इतना बड़ा आपका mRNA तो इतना बड़ा ही आपका polypeptide chain होगा. Your mRNA can have open reading frame from here till here. Okay, it just depends on the sequence it is carrying. वो सिर्फ information है जो carry कर रहा है. Rest the length of the polypeptide chain determines is by that information. So open reading frame सभी को मालूम है. Okay, so open reading frame everyone knows that it is starting with your start codon, which is your AUG. Correct? Now there was this question why AUG coding methionine is always the first codon. Can anyone give me the answer? I repeat my question. चाहे prokaryotes हो, चाहे eukaryote हो, okay? Always your first codon is your methionine. Why? The only difference comes in the formal methionine and methionine, but why it is always methionine? Yes, क्या हो सकता है आंसर? सिंपल एक लॉजिकल आंसर क्या हो सकता है इसका? यस, अक्षया, श्रुति, तन्वी, कुछ भी आइडिया सोचो क्या हो सकता है? Simple. Because it is coded by the first codon. जब evolution ने decide कर लिया है that AUG should be your first start codon and AUG codes for methionine, then obviously it has to be your first amino acid present in the polypeptide chain. Correct? And whatever the first initiator tRNA जो आपका रहता है, initiator tRNA which brings the first amino acid, okay, it is also carrying the methionine. So these are the ways by which you can give the answer. Is it clear? It was just twisted so that you guys can think more on it. अगर आप ये पूछो कि क्यों एवोल्यूशन ने AUG को ही डिसाइड किया है फर्स्ट कोडॉन लेने का, then you can argue the question, argue the statements. Since this has been decided by the evolution that AUG should be the start codon in the mRNA and AUG codes for methionine. अगर AUG उस टाइम लाइसिन को करता तो आज लाइसिन हमारा फर्स्ट कोडॉन होता. But since it is coding for the methionine, so it is methionine. Your first amino acid is your methionine. Now it is clear? Yes, Tanvi, able to follow?
Yes, ma'am.
Okay, okay. Shruti, clear hai? Akshaya?
Yes, ma'am.
Okay,
I
okay.
thought that the question was why AUG was a start codon.
No. Then you need to ask to the evolutionary people that why AUG is the first start codon. Why they have taken. See, मुझे क्या लगता है the answer would be AUG नहीं होता तो let us say that UU होता. ठीक है? Then it would have coded for the phenylalanine. But I think the rest is going to be the same. आपको कहीं से तो कुछ तो शुरू करना है. So the evolution has started taking AUG as the start codon. I feel in that way. कहीं से तो शुरुआत करनी थी. If not AUG, then triple U, whatever. So then the scenario will be different. Right now it is AUG coding for the methionine. That's why it is the first codon. I believe this is a thing. आप कोई भी चीज लो, the starting, it needs a starting point from somewhere, right? So that's what I believe. Like जैसे evolution जब हो रहा था, translation was coming into the picture. What I feel is that time which was mostly available for the translation machinery that time. So in that way they have adopted. Okay, कि अभी हम लोग इसी से शुरू कर लेते हैं, next round भी इसी से ही होगा. So it gets embedded into the evolution. Okay, and we like the evolution says that eukaryotes have evolved from the prokaryotes. So when prokaryotes have evolved in such a way that they have taken AUG as a first start codon, obviously the eukaryotes even have the same first codon. Then जैसे जैसे changes आते हैं, diversity आती है, then we have got the differences in the stop codon. So there are now the codons are there, like that's why it is universal, right? The genetic code is universal. Now later we have seen that the mitochondrial and the chloroplast genome, they code for something else. So there it becomes difference because you know how chloroplast and mitochondria have evolved, right? So that's why there is little differences in those codons over there. But this is what I believe. मैंने इससे ज़्यादा और कहीं कुछ पढ़ा नहीं है. And I haven't got also, like this was really a very interesting topic for us also to discuss, but as such more information even I didn't get. So in future if you guys get, please share in the group also with us. Okay, so the translation starts at the 5' end of the ORF and proceeds one codon at a time to the 3' end. Okay, so once it is read in 5' to 3', in the case of protein it will be known from N terminal to C terminal. Okay, the first and the last codons of an ORF are known as the start and the stop codons. In bacteria, the start codon is usually AUG, but 5' GUG and sometimes even 5' UUG are also used. So ऐसा नहीं है कि in the case of the bacteria it's always AUG. Depending on the species variety, the start codon can vary. Okay, but in the case of eukaryotes, it's always the 5' AUG. Okay, so I believe this is simple and all, nothing, no confusion is there. So this is again your start codons is AUG, it is coding for the methionine. And there are 3 stop codons, UAA, UAG, then UGA. I mean, that's how I remember. Now, can you tell me among this, which is amber, which is opal, which is, or kaun sa tha? There are 3 names of these stop codons also, right? Yes. So what are these names? I mean, I'm sure nobody's going to ask this. This is very basic, but I believe that you should know all this thing. So this is known as your amber. This is known as your ochre. And the last one is your opal. ठीक है, ये उन लोग के नाम के ऊपर है जिन्होंने ये डिस्कवर किए थे. Clear? Simple है, कुछ इसमें याद करने जैसा है नहीं, but yes, like even I do get confused with these stop codons. So what I do is I just remember that UAA is stop codon, फिर यहाँ पे G and then इसको हम shuffle कर देंगे, then also I'm gonna get a stop codon. So that's how I remember. Otherwise, even I'll get confused. Okay. Now, so the start codon has got the two important functions. So first one, it specifies the first amino acid, of course, to be incorporated into the growing polypeptide chain. So इससे क्या होगा? I'm gonna get something onto which I can start adding the nucleotides now. Okay, so I have started my protein synthesis. Second, it defines the reading frame for all the subsequent codons. Now, the beauty about the codons is that already discussed, it is not overlapping, right? And it is continuous, and there is no comma. तो हम कैसे लिखते हैं, कोई भी अगर हमें कोडॉन्स मिलता है तो AUGA continuously we will be writing like this, right? So the mRNA or the tRNA, they read in the 3 nucleotides regions. Now अभी इसी में आपको एक चीज क्या समझ रही है, what if A is getting deleted, what will happen then? What will happen to the ORF? What will happen then? Yes. Yes, Akshaya, what do you think what will happen if suppose A is getting deleted? And I'm saying that your ORF starts from here only, AUG. Now, while the ORF has been determined and because of certain reasons, A got deleted. अब क्या होगा?
The translation won't start because AUG is the one which starts the translation.
Correct. And what will happen? Now, the mRNA will be read as starting from UGA. Okay, it will be taken like this, UGA, AGU, and so on. And what is UGA?
Stop codon.
It's a stop codon. So, what do you think? The translation is not going to occur there. Okay. Okay. Now, once the translation starts, however, the reading frame is determined. Thus, by setting the location of the first codon, the start codon determines the location of all the following codons. Now, this is also important when you know when you are going to do the research. So, कैसा रहता है? You are going for the molecular cloning, correct? जिसके लिए आप primers बनाओगे. Now, once you get the primers, you are going to get your gene also. ये gene जो आपको मिलेगा, you are going to do the restriction digestion. You are going to remove from the vector. Okay, so vector तो हमें नहीं चाहिए. I just want the gene. Now, gene I'm gonna take. Now, I need to confirm whether the gene is correct or not. So, what will I do? I'm gonna give it for the sequencing. Okay. Now, one nucleotide base pair बनाने के लिए they charge 13 rupees. ठीक है, to sequence. तो जितना बड़ा आपका जीन है उतना चार्जेस आ जाएगा. सो इफ यू हैव गॉट 500 बेस पेयर का जीन into 13 will be the charges plus GST whatever it is. तो ऐसा है. अभी तो मार्केट में 13 रुपीज है. I don't know if recently they have changed, कम किया ज्यादा किया, I don't know. So it looks cheap, 13 rupees, but you know that the genes are always more than 500. And then you also want that it has to be accurate. So आप उसको 3 बार कराते हो फिर minimum 3 rounds of replication. 3 rounds of sequencing. So that's how your charges gets increased. And of course सिर्फ आप ही का sequence के लिए नहीं जाएगा ना, और भी lab में members होंगे, इन सबका एक साथ जाता है. So then once I get the sequence, they're gonna give me the sequence. Okay, I'm gonna get the sequence. Now there is some softwares are there, like chromatogram is there. So I'm gonna upload my sequence over here. Now the sequence they are going to give me, A, T, G, C and all I'm gonna get. Now I need to check from where my gene is starting. मुझे मेरे gene का start codon, stop codon ढूंढना है. ठीक है? So many softwares are there, but usually if you are doing for the first time, then they try that you actually go manually. Okay, जरूरी नहीं है हर lab में हो, but yeah, they might give. So there are softwares, you can go to the NCBI also, where the gene what you get is the DNA sequences, correct? What you can do is you can convert them into your CDS, coding sequence. So उसके लिए software है. What you can do is you take the FASTA format of your sequence. It's called FASTA. Okay. Please remember all these because हम जब bioinformatics भी करेंगे, the important topics, all these we are going to study the FASTA format. Okay, so the FASTA format you're going to take, and then you're going to put into the software. The software is going to convert your DNA sequence into the CDS. What is CDS? The coding sequences. Okay. Now, from here, CDS, you're going to search for your start codon. Okay. Now the important thing, what you need to check. हम करें क्या इस sequence का? हम करेंगे क्या इस start codon का? Now, let us say that. Let us imagine that this is like from here to here. Let us imagine that these are your sequences. Just imagine that these are your alphabets, okay? जो आपका codons में आएंगे. So S E C, I'll take one. O N D, I'll take it one. Just imagine like that, okay? अब मुझे ये check करना है कि मेरा start codon कहाँ पर आ रहा है. So obviously it is not going to start from here. Somewhere here it is going to start. Let us say that it is starting from here, okay? Happy. Okay, I got my first AUG. AUG तो नहीं, वो ATG दिखाएगा, ठीक है? Okay, so I'm happy that I got my start codon. But what if I'm getting stop codon here itself? Immediately, I do have this much big gene is there, correct? But my start codon is here, stop codon is here. Do you think it is reasonable to clone this particular gene? Yes. What do you think, guys? Agar, are you able to follow me? Yes, are you able to follow? If I have start codon over here and stop codon over here, do you think it is reasonable for me to go further in the cloning? तो उससे क्या होगा? Now I know that I'm gonna get a truncated polypeptide or truncated protein I'm gonna get. Correct? So now I know with the help of the bioinformatics tool that it is going to give me a truncated protein. तो मैं क्यों मेरा time gene cloning में करूँ? So what should be the next step if I really want to work on this gene? तन्वी, क्या लगता है क्या करना चाहिए फिर मुझे? श्रुति?
Maybe you can try to get another. You can try to isolate it again.
Yes, either something's wrong with the gene, so you can go for the gene isolation again, or maybe from the different organism, but having the same function. Then next step, what you can do is, suppose आपको यही मिल रहा है, but you really want to make sure that this gene is working, ठीक है? Either you change the gene, or you can go for SDM. What is SDM? Site directed mutagenesis. ठीक है, तो मैं क्या करूंगी? स्टॉप कोडॉन जो है, यहाँ पे कोई सा भी स्टॉप कोडॉन आ रहा है, सपोज लेट अस से दैट इट इज कमिंग योर UAG आ रहा है, ठीक है? सो व्हाट आई कैन डू, आइदर A चेंज कर दूंगी या G चेंज कर दूंगी, ठीक है, जिससे मुझे स्टॉप कोडॉन की जगह कुछ और अमीनो एसिड मिल जाए व्हिच विल कोड फॉर समथिंग एल्स, एंड आई विल रिमेंबर दैट आई हैव चेंज्ड दिस सीक्वेंस. ओके, सो देन आई विल सी व्हाट काइंड ऑफ प्रोटीन आई एम गेटिंग. So we can do such things. And मैं यहाँ पे, अगर यहाँ पे कोई भी कोडॉन नहीं आ रहा है, so I can insert that particular thing. अगर suppose U था, so I can just change, right? I can just change one simple nucleotide so that I can get a stop codon at the end over here. So this is how site-directed mutagenesis is helping in designing of the genes. Okay, so these are the ways by which you can do. Okay. Now translation begins at the start codon. So you have got the mRNA coding region, you have got exon 1, 2, everything. Now what are the possible ways by which you are going to have the mRNA codons? So these are the three possibilities, because we have got the codons read in 3 nucleotides, right? So एक हो सकता है जहाँ पे वो UAG से शुरू होगा. You know that UAG आपका stop codon है. So obviously this mRNA is not going to be functional. Next case, if ये UAG उन्होंने consider किया, यहाँ से they have started reading like this. Suppose the open reading frame reads from here, AGA, GUC, then AUG. So now I got my start codon. So it is going to have the mRNA. But again, you can see that stop codon is immediately coming. So if I have the requirement of having only 1, 2, 3, 4 peptide का अगर मुझे requirement है, then तो this mRNA is going to get translated. Otherwise, this will also get degraded. Now look at over here. What is the another scenario? हमने UA छोड़ के G से शुरू किया है. GAGUCA से बोलना शुरू किया है. What do we see? That I got only stop codon and there is no start codon. So obviously, out of all these three possibilities, only one is giving you some output. Okay. So the nature is going to select this particular open reading frame for your translation. Is that clear, guys? Yeah. Is it clear?
Yes,
Yeah.
Okay. Now ORFs. So mRNAs contain at least one ORF. Now, in the case of bacteria, you can have multiple also. So the number of ORFs per mRNA is different between the eukaryotes and the prokaryotes. Eukaryotics always contain single ORF, whereas in the case of prokaryotes, you can have multiple. And those are known as polycistronic mRNAs. Okay, so we'll see their translation. There's a little difference, like how the prokaryotes and the eukaryotes carry out the translation. And the then if you have got only single ORF, then it is your monocistronic mRNAs. Okay. Now, polycistronic mRNAs, which is found in the bacteria, नाम से ही पता चल रहा है poly है. So what does it mean? The single mRNA will be coding for different polypeptide chain. Okay. So what does it mean? It means that the single mRNA is going to have start, stop, start, stop, start, stop. Okay. So this means what? How many are there? There are 3 cistrons are there. So 1, 2, 3. So I'm gonna expect 3 polypeptide chain. Clear? If it is only 1 start and stop, it is monocistron. If I have got 3, then I can have 3 cistrons and 3 polypeptide chain. Okay? Yeah. Look at the figure. Beautifully they have shown. Now, if suppose this is your mRNA, you can see that there is no cap over here. Can you see? And what they have shown is 16S rRNA. So we know that in the case of prokaryotes only, the ribosomes 30S and 50S is there, if you remember, right? The smaller subunit and the larger subunit. 16S rRNA is part of your 30S subunit. So ये figure देखते ही आपको समझ जाना चाहिए that they are talking about the prokaryotic mRNA, and below they have not mentioned anything. But on what basis can you say that this one is belonging to eukaryotes? Even though it is not mentioned, but looking at the figure, you should be able to pinpoint. So कौन बता सकता है that why it is eukaryotes mRNA? Because of the presence of dash and dash. Yes, Akshaya.
Single start and stop region, so there is only one mRNA formed at a time.
No, that is not the reason. ये तो मैंने just अभी आपको बताया है कि that eukaryotes code, I mean they have got only one cistron, but there is something else which is not present in prokaryotes.
The 5' cap and the poly A tail.
Correct, absolutely correct. So 5' cap and the poly A tail. This is only present in the case of eukaryotes. Okay. So from this you should be able to understand कि नीचे वाला eukaryotic mRNA के बारे में है. Okay. Now coming back to over here. So what do we see? That ये तो हो गया. चलो. Now what do we see over here? That you have got start, stop. Okay. Again start, stop. Again start and stop. So it is going to code for your 3 polypeptide chain. Now, I don't know whether you have studied or not, but whenever we talk about bacterial operons, lac operons कभी सुना है? lac operon, which is for the lactose, so that the bacteria are able to metabolize lactose whenever in the media glucose is not available. Have you heard about the lac operon? Suna hai? Anyways, we are going to do this in detail, but have you heard of? Yes. So lac operon bhi aise hi rehta hai. So it is going to code for your, we have got Z, Y, X, all those, sorry, ulta likh diya, Z, X and Y, we have got those genes present, which is going to code for these enzymes like lactose permease, beta galactosidase, all these enzymes, it is going to code so that the bacteria is able to metabolize lactose. Lactose is actually a disaccharide which is made up of galactose plus glucose. Okay, so जब मीडिया में ग्लूकोज अवेलेबल नहीं होता है, then bacteria, glucose is your monosaccharide, readily available energy source, right? Now, if in the media glucose is not available, lactose is present, then bacteria should be able to metabolize lactose. तो उसके लिए आपको separate enzymes लगेंगे, which can degrade your lactose into galactose and glucose. So, of course, once it is degraded, glucose can be taken and galactose, with the help of the epimerase, can be converted galactose to glucose. Okay. So, अगर आप देखोगे galactose और glucose में, it is actually a 4-OH epimer है. तो epimerase enzymes होते हैं जो galactose को glucose में convert कर देंगे, and eventually the bacteria will be able to use the entire lactose. Okay, that is different part. Now, the same concept of over here polycistronic mRNA will be used when we will talk about the lac operons. Okay, so this is one point. Now there's something. Okay, anyways, this diagram is going to repeat. Now coming to the eukaryotes. Now, in the case of the eukaryotes, you can see that pi dash cap is a, poly A tail is a, and you have got the start codon and the stop codon. एक और चीज आप ध्यान दो. Every time, whenever we talk about start codon, it is not always starting from the very first nucleotide, right? You can see it is not always the first nucleotide. ये हमेशा आपका कहीं बीच में से ही आपका AUG जो होगा वो शुरू होगा, and from there onwards your first amino acid will be coded. Okay. Now, why it is so? Same, because it needs to bind to the ribosomes. It needs that arrangement on the ribosome so that it can start coding for it. तो वो base जो है वो आपको ये region provide करता है. Okay. Now, if you see in this figure also, you can see that there is something called RBS. RBS में कुछ ऐसे sequence है which is able to interact or which can form complementary base pairing with the 16S rRNA of your 30S subunit, right? So these are the regions which make sure that, okay, let us get aligned properly so that the translation machinery is ready, and then they can start for the translation. We'll come to that. What is RBS? Okay, this is another way of representing the same thing. So you can have the multiple translation sites. So you can see untranslated region, start stop is there. In between, you can see those gaps. So those gaps are required so that the translation machinery are properly aligned, so that it can code for the proteins. Okay, so एक ही mRNA में 3 polypeptide chain can be synthesized. In the case of eukaryotes, you can have only single one. Okay, now the important part. Prokaryotic mRNAs have ribosome binding site. जो अभी हमने previous figure में देखा था RBS which we were talking about. Okay, so that is your ribosome binding site. Okay, so RBS now, right? So RBS is your ribosome binding site. Now for the translation to occur, ribosome must be recruited to the mRNA. So this information is present where? It is present in the mRNA itself in the form of your RBS. So this is what RBS stands for. So as you can see that before your start codon, 3 to 9 bases above, okay, it is present, the RBS, ribosome binding sequences, and GGA, GG. So these are the sequences to which complementary your 16S rRNA is going to base pair. This is also known as Shine-Dalgarno sequences. ये थोड़ा सा याद करना पड़ेगा because everywhere it is not written as RBS. It might be represented as Shine-Dalgarno sequences. Same thing. Now, the RBS base pairs with this RNA, aligning the ribosome with the beginning of the ORF. So अभी कैसे होगा? ये mRNA हो गया. When it is represented 16S rRNA over here, so what does it mean? यहाँ पे आपका 30S subunit है. Obviously, in the opposite direction, you are going to have the 50S subunit. Similarly, in the case of eukaryotes also, we have got certain sequences which make sure that the ribosomes are coming and binding to those particular regions. Okay, so in the case of eukaryotes, it is known as Kozak sequences. याद कर दिया, so it is good, well and good. Okay, you can see GGA-GG. So obviously यहाँ पे कैसा होगा? It will be CCU-CCU. Correct? Opposite. You just have to remember in that way. Complementary. Okay. Now, translation coupling. This is also important. So right now, what we have seen was about the polycistrons, right? In the case of prokaryotes, we saw polycistron. So, but in the case of the polycistron, each cistron has its own start codon and the stop codon, correct? And the RBS unit was present. If you have seen in the previous figure, RBS start and stop was there. In few cases, okay, prokaryotic ORFs, they don't have strong RBS, but still they are actively translated. Okay, so in this case, ORFs are not the first ORF in an mRNA, but they are instead, they are located just after another ORF in a polycistronic message. So what does it mean? In this case, if you have got one RBS, start codon, followed by stop codon, and immediately you have got the start codon. Okay, then you are going to have the stop codon. So इसमें क्या होगा? The same ribosome, okay, the same ribosome is going to continue here your translation. Are you able to imagine? I'll show the figure also, don't worry. So in these cases, the start codon of the downstream of ORF often overlaps with the 3' end of the upstream ORF. Thus, a ribosome that has just completed one of the translation of the upstream ORF is now positioned to begin translating the start codon of the downstream ORF. So in this way, it is just doing the continuation, and this is known as, because of these overlapping ORFs, it is known as translation coupling. Okay, the concept of translation coupling is clear, all of you. I'll just show the figure. So it is important to note that in this situation, translation of the downstream ORF requires translation of the upstream ORF. So what will happen if suppose the upstream ORF is not able to get translated, then obviously the downstream ORF is not going to happen. ठीक है? तो अगर मैंने इसको कैंसिल कर दिया तो डेफिनेटली ये भी आपका नहीं होगा. अगर ये अच्छे से हो रहा है, then definitely ये अच्छे से होगा. Simple. Okay. I'll just show the figure. I think it will be clear then. Now, as you can see in the figure now, so you have got the ribosome coming over here. This is your larger subunit. This is your smaller subunit. mRNA hai. It has got its stop codon over here. This is just the mention. Okay. Now, this stop codon is again followed by the another. Okay. Now, what is happening? You can see that the moment ribosome reaches the stop codon of this particular ORF, it is now continuing the translation of the second cistron. Clear? So this is how it happens in the case of your translation coupling. This is your translation coupling. So you have coupled two translations together without the need of RBS. Clear, guys?
Yes, ma'am.
Okay, simple concept. The only thing is like how the questions will be asked, that is more tricky. So we'll do questions also, don't worry. Now coming to the eukaryotic mRNAs. So eukaryotic mRNAs are modified. So as right now we have discussed, in the case of eukaryotes, we have got the 5' cap is there, and how 5' cap is made. So there are two enzymes required. अगर आपको याद हो, हमने discuss किया था, there are two enzymes required. एक guanine आपको recruit करता है, and then you have got the methyl transferase, which adds a methyl group. And at the end, at the 5' end, guanine में ही आपका 5' to 5' linkage बनता है, if you remember. Okay. Now what happens is that ribosome, in the case of the eukaryotes, they will scan for this 5' cap. And once they get this 5' cap, then they start reading your start codon. So that particular process is known as the scanning. So इसलिए, see, in the case of prokaryotes, it was the RBS sequences which was scanned by the ribosomes. Okay. Now once the RBS, the Shine-Dalgarno sequence they get, the ribosomes get properly aligned and the translation happens. Now in the case of eukaryotes, the scanning which is done by the ribosomes, it is for the 5' cap. जैसे ही 5' cap मिलेगा, they get aligned over there. Of course, there are other translation factors also involved. They come together, they scan the mRNA for the AUG. That is a start codon. Okay, so this is how the translation initiation is happening. So what happens then? The smaller subunits, it is going to scan for the methylated cap. Once it gets the cap, then it is going to migrate so that it can understand where the AUG start codon is present. Okay, so this is the difference between the your prokaryotic and the eukaryotic sequences, like how it is going to start. Now there is another features in the case of eukaryotic mRNA which is helping the translation. Okay, stimulating the translation, यानी कि क्या translation को enhance करने में help करता है. So there are again like RBS, we do also have certain sequences known as Kozak sequences. So what are these sequences? These sequences are like, if you see, यहाँ पे देखो sequence की तरफ देखो, you have got AUG. This is your start codon, right? So usse pehle you have got these three sequences which is known as Kozak sequence. So what this person, okay, Merlin Kozak has found that whenever he was working on these eukaryotic mRNAs, he always found that before this starting of the codon, always these three sequences are present. Basically, those are ACC. ACC. So he saw that around 95% of the eukaryotic mRNA, whenever there is a ACC sequence, it is always followed by the AUG. Okay. So this sequence is known as your Kozak sequences, which is found conserved throughout the eukaryotic mRNA. And it also says that these bases are thought to interact with the initiate tRNA and not with the RNA component of the ribosome. So can you see the difference? So in the prokaryotes, we saw that the Shine-Dalgarno sequences said that it is for the ribosome interactions. Okay. So these RBS or the Shine-Dalgarno sequence, they interact with the RNA component of the 16S RNA, that is a part of the 30S ribosome, right? वहां पे आपका prokaryotic mRNA start होता है. Whereas in the case of the eukaryotes, he found that it is always to interact with the initiated tRNA rather than with the ribosome. So this is the difference, and always before the start codon, he always found that ACC is present. Okay. So these are the plot which tells you like, अभी देखो यहाँ पे देखो, can you see the capital A, फिर G आ रहा है, फिर आपको the size of C, G, A and ये से, and here you can't see the this. What does it mean? Like how to read such graphs? What does it signifies? अगर कल को अगर आपको ऐसा ग्राफ मिले, how you have to read is, this is for your ATG sequences, right? So the maximum जितने बार जितना like simple way है, जितना बड़ा letter होगा आपका alphabet जितना बड़ा होगा, it means the occurrence of that particular nucleotide is high. Okay, for example, at this position AG का होना बहुत ही high है, and then C का उतना है, and then over here C का. So this percentage it will tell you. Okay, so what this particular thing is saying that at 11, 12 and 13 ATG तो confirmed है, it is throughoutly it is present. But when it is checked with the other eukaryotic mRNAs, so the positions were occupied by either A, G, C, C and C. So basically what you have seen is that ACC ka presence was more as compared to the rest of the nucleotides before AUG. Are you able to follow, guys? And then you can have the other sequences which is coding for the codons. So AUG to ho hi gaya hai, and then you can see the G is more prominent, right, as compared to the other alphabets like A, C, T over here. So iska matlab kya? Maximum time ab jab bhi codon sequence dekhoge, so after your AUG always G will be present, and isse pehle hamesha ACC present rahega. So कोई भी mRNA sequence में हमेशा हमें ये जरूर दिखेगा ACC followed by start codon followed by G, and then you can have whatever codons are present over here. So this is how you read such graphs. Able to follow?
Yes, ma'am.
थोड़ा सा difficult जाएगा, but with practice I guess you can do, because this is how the chromatogram you're going to read also. I'll just repeat once again short form में. So this says that 11th and 12th, 13th position, it is ATG. This is DNA sequence they take. Okay, so ATG is present over here. Okay, now they say that in rest of the cases, they have seen that after ATG, the probability of G is high as compared to the other sequences. ठीक है, तो यानी कि इसके बाद G होगा. Then what do we see? I can see that C is present over here, right? And there is no other. उसके नीचे A आ रहा है, तो इसका मतलब क्या? C के बाद the chances of A is high. Okay. अभी हम इस साइड आ जाते हैं. Over here, the chances of A is maximum at your 8th position. इसके बाद C है, इसके बाद C है. So अगर मैं in future कोई भी codon sequence या coding sequence लेती हूँ, I'm gonna get ACC, then AUG followed by G. Okay. So this much तो मेरा fix रहेगा, depending on the, अगर मेरे पास कुछ बहुत ही distantly related organism है, then I might have certain other nucleotide over here, but this is mostly conserved sequences. Okay. So ये experiment से हमें ये पता चला है. Okay. So a second feature that contributes to efficient translation is the presence of the poly A tail at the extreme 3' end of the mRNA. Now, this is also believed to enhance the recruitment of the key translation initiation factors. So once all these recruitment with the help of the 5' cap and poly A tail happens, then your translation initiation complex is ready. Okay. So ये इतना आसान नहीं है. There are many things, many factors required in which the translation is going to start. Okay, so far so good, guys. Able to follow?
Yes, ma'am.